So today we're going to discuss histone modification. Let me see, where is my, okay. Okay. You know, on this slide, I show you the structure of nucleosome at the bottom. You know, that's the, EM structure, or it's just an illustration of the nucleosome. The nucleosome is the fundamental structure of chromatin. So basically, you have 146 nucleotide wrapping around the one histone octamer. The histone is an octamer, right? So it's a protein. It's a protein core there, and then you have 146 DNAs, 146 nucleotides wrapping around for about 1.7 ROMs. Okay, that's the fundamental structure of nucleosome. And on top, I give you the illustration of the four histones. So the histones is the octamer. Basically, you have two copies of each of H2A, H2B, H3, and H4. You have two of each, and therefore you have an octamer there. So you can check your textbook elsewhere to refresh yourself about the histone structure. But today, I just want to point out that you can observe the illustration on the top panel. Each histone will have a so-called histone fold. That's the common sequence, the common domains of all the four histones. And then for histone modification, we care mainly about the N-terminus, because all of those histones, the four histones, have a N-terminus there, and with H3 have the longest N-terminus. For H3, you even have an extra domain there, right? An extra N-terminus domain as shown there. Well, the rest doesn't have that specific domain for H3. And all the modifications, I should say, the majority of the modifications of histone happens on the N-terminus. And here I just want to show you from the bottom panel again, what's that N-terminus on this illustration. You can see the dangling part showing in green color. That's histone H3. The color code is exactly the same as in both illustrations. So that green part is the N-terminus of H3. You can see it is dangling. It's dangling there. It's not like other histones. The other part of the histone is, that could be within the core, but this part is free floating. So any modification could have the potential to mediate some change. Like a modification could affect how two nucleosomes are associated with each other, because this floating part, the free floating part, may be interacting with each other, right? So if you have a modification there, it could disrupt the interaction between two nucleosomes or mediate the interaction between two nucleosomes. So both can happen to both directions, either association or disruption of an association. That could be one consequence of modification, or it can affect how DNA and the histones are interacting. A modification could all of a sudden make the DNA and the histones not interacting as well as originally, or the opposite. So all the modifications are related to how nucleosomes interacting with each other, how histone and DNA interacting with each other. Okay, so we're going to today we're going to discuss the several modifications and their significance, their biological significance. But on this slide, I also have additional information of of the most fundamental epigenetic regulation. As we have already covered, the DNA modification apparently is one of the most important regulation. The second will be histone modification, which we will cover today. And the third aspect we will not cover is interaction between them. Okay, but keep in mind all those could contribute to the change of your chromatin structure, and then consequentially the change of gene expression. So here I show you the 4 histones again, H2A, 2B, H3, and H4. And the basic types of modifications would be phosphorylation, acetylation, ubiquitination, and methylation, as shown on this slide. And all the sequence on this slide are the N-terminus. The structure the sequence starts from amino acid 1 and then go all the way to somewhere, so that remains to be the N-terminus. We're going to discuss mainly about acetylation and methylation today, but phosphorylation and ubiquitination can both occur, although the latter two are not as widely studied as acetylation and methylation. But I would like to point out that phosphorylation, acetylation, and methylation involves a very small footprint, right? The phosphoryl group, the acetyl group, or the methyl group are small relative to ubiquitin. Ubiquitin is quite big. Okay, it's a big protein there. So the modification by ubiquitin is kind of different from the rest three. And again, as you can see, that phosphorylation and acetylation will carrying negative charge, because either phosphoryl or acetyl will be negatively charged. And these modifications often happen on lysines. As if you observe, you see that a lot of those modifications are on lysines. The lysine have a long side chain. Remember, it's like 4 copies of CH2, and at the end you have an amino group. So it's a long arm there, and then ended up with a positive charge. So that's phosphorylation and acetylation will neutralize that negative, that positive charge, making that part to become negative. That's why the phosphorylation and acetylation can carry in some major changes, because previously maybe there are some electrostatic interaction mediated by that lysine side chain, positively charged lysine side chain, and now that modification will just abolish that interaction because the charges are all of a sudden very different. Of course, you can tell methylation and ubiquitination will not carry that effect. So the methylation and ubiquitination probably mainly is because of their the structural change they were introduced. Well, acetylation and phosphorylation can change the charges. And I guess the one thing I emphasized is the modification usually occur on lysines. This, lysine is a strange residue and also a very important residue. You learned previously that lysine can be involved in catalysis. It's a covalent catalysis, right? Because I believe shift base formation often involves a lysine. And the lysine also contribute to a side chain of peptide, because the lysine have a amino group. It can get linked to another polypeptide. Therefore, the protein is not a linear chain any longer. You have a branch. The branch out often happens on lysine. And also, lysine is involved in, interacting with cofactors, such as biotin. We learned that in biochemistry that the long arm of lysine will be linked to the long arm of biosine. Therefore, that is involved in carboxylation. So the lysine is a very important residue. I hope when you gradually learn in different subjects, you try to put your knowledge together to appreciate a bigger picture instead of getting everything to be fragmented. That's just not the best way to learn. So here, I would like to reiterate this chip sequence technology again. So the full name of a chip is chromatin immunoprecipitation, and this so-called chip is often followed by sequencing, DNA sequencing. And this is such an important technique in epigenetics, especially histone modification and transcription factor binding. Therefore, I would like to explain that process again. So shown on top is just the fundamental structure. It's a nucleosome and sorry, it's so-called the bead thread and bead structure. So you have nucleosome, that's your beads, and then thread is your DNA. As we have explained earlier, if the DNA is wrapped around the nucleosome, and usually the gene there is not active, so naked DNA turn to get expressed. So usually transcription factor will bind to the naked part, so like shown here, the green oval shaped thing is just a transcription factor there. It will bind to the naked region that indicate a transcription factor. Well, in B, the other column there, the red color indicate a modification of histone. As we have learned, this dangling part, perhaps it's histone H3, and now the N-terminus have some kind of a modification, and that modification is indicated as red. So our task here is to identify that modification in B, or in other occasions, you want to identify the transcription factor that bind to a particular region. So CHIP is the method of choice for these two tasks. So usually the procedure starts with cross-linking. The cross-linking basically will fix that protein to DNA, no matter it's a transcription factor or histone. So that interaction, the protein to DNA interaction is now fixed. They won't be able to get dissociated. So this is called cross-linking. And then in the next step, what you do is fragmentation. So why do you do a fragmentation? Because eventually you want to pull a short fragment of DNA down, not the entire chromosome. If you pull down an entire chromosome, you're not identifying anything, because no matter what modification you have, if you don't do fragmentation, you're going to pull down the whole chromosome. So then which part has that modification, you cannot tell, because the resolution is now so low every time you just pull down a chromosome. So that's why you do a fragmentation. The fragmentation can be done with either enzymes such as endonuclease, or you can do it by sonication. Sonication can break the chromatin to fragments. And in the next step, what you do is using an antibody to capture that protein if it is a transcription factor. Like shown on the left side is a transcription factor. You have a specific antibody that goes against that transcription factor. You use that antibody to capture that protein. In histone modification, you have a specific antibody that can detect that modification. For example, like a histone acetylation. And now there is a protein that is specific for acetylated histone H3 on a particular lysine, like lysine 27, for example. So you have an antibody that's so specific that can recognize that modified histone, and now the antibody can capture that protein. And then in the next step, the antibody, you do a precipitation. basically the antibody will pull down the protein, the transcription factor, and the associated DNA. In histone modification, that antibody will pull down that histone, the modified histone, together with the associated DNA. And eventually, you identify that associated DNA by sequencing usually. So the result will be identification of DNA sequences that is associated with that particular modification. So eventually, what you get is DNA sequence. Now, you put that DNA sequence, you do an alignment throughout the genome. Now you can tell, okay, maybe this chromosome 3, a particular region have a transcription factor. Or you can say it has a modified histone. Okay, I hope you can appreciate this, because if you don't appreciate this technique, we lose all the purpose of explaining histone modification and DNA, or even DNA methylation. So now, our country have already developed some consortium, some database. So a lot of information has already been collected. So this famous project by the U.S. National Human Genome Research Institute, established this project called the Encyclopedia of DNA Elements, or in short, ENCODE. They have this project, then they captured a lot of information in addition to the Human Genome Project. Previously, the country have already sequenced many individuals of their DNA sequence. So the DNA sequence, the genomic information is known. The DNA sequence is known for a lot of individuals. And now in this ENCODE project, they try to capture epigenetic information. As shown here, I'll tell you what information they have collected. For example, we start with RNA sequencing. We start from the right, that's RNA sequencing. They have studied what transcripts are there. So the DNA sequence are all the same. For most people, it's the same sequence, right? But sometimes in different ethnic groups, you could have some variant. By sequencing human genome, you're identifying all those variants. Otherwise, the sequences are quite similar to each other because we are all human beings, right? So you have the same sequence, generally speaking, but there will be some variant. But now in this ENCODE project, they do RNA sequence. That means, okay, in your brain, in your heart, it's going to have different RNA expressed. In this ENCODE, they capture that information. In this disease versus another disease or normal person, RNA expression are different. They capture that information. They have also done a lot of ChIP-seq. In this ChIP-seq, they identified modified histones, where the histone modifications are. They also identified the binding site of transcription factors. Like I explained in the previous slide, you can identify the transcription factor binding site if you have specific antibody against the transcription factor that you're interested in, right? If you have an antibody, you can perform check. You can identify where this transcription factor bind throughout the genome, because transcription factor will not bind to only one place, will bind to many places, regulate many genes. You're trying to identify the spots where those transcription factors will go throughout the genome. And of course, they use the technique called DNase hypersensitivity. I explained it in the previous lecture. And the Fairy sequencing, I also explained it in the previous lecture. That's about which region is more relaxed, which region is more compact in terms of the presence of nucleosome. Okay, that's what we explained previously. But then they also performed something called 5C. I couldn't even remember the full name of 5C, but because there are too many names. They have 3C, 4C, 5C, 6C, 7C. So I lose track of I lost track of the names. But all those C's means they're trying to identify even higher level structure. And look at the rest. We have just covered, no matter it's DNA's hypersensitivity, ChIP-seq or RNA-seq, everything is linear information. Okay, you're trying to think that the DNA is just a linear strand. Therefore, the binding, for example, like here, if you have this pink ball, which is a transcription factor, binding to this negative region, you would start to predict maybe the adjacent genes will be regulated by this transcription factor. That's your conclusion, but that conclusion is incomplete, to say the minimum. It's incomplete because throughout our genome, the organization of genes, the organization of nucleotide, the organization is not linear. Look at this region. You could have, the chromosome goes as linear and then wrapped back. Therefore, a transcription factor, if it is located here, it will not only affect the adjacent gene, it may affect some other genes that get wrapped back. Okay, because the structure apparently is 3D. The DNA is never linear. It's packed in our nucleus. Therefore, you can't just say that this transcription factor is located in this A location, therefore only the genes around that A location is affected. It could be like several megabase away, some genes are affected as well because everything is 3D. In 3D, in terms of 3D, maybe the distant part is actually quite close. Understand what I'm saying, right? Because linear-wise, it's very far away, but if you think about the structure in 3D, that part could be very, very close to this transcription factor. Okay, so they're trying to capture that information as well, but apparently that information will be even more complex, very difficult to appreciate. So we have to, go with our limit. So somebody who with advanced equipment and better mathematical models eventually will start to appreciate the 3D structure. But at this time, as in this lecture, what I explained to you is still linear information, which, as I said, is incomplete. So here I show you one plot from the ENCODE project. So basically, you have the DNA sequence, it's the gene code genes, right? And you have a DNA sequence, therefore, you should be able to predict throughout our genome where you have this P53 gene, where you have this RB gene, where you have CDK4, where you have P27, all the genes you should be able to tell where they are. That information should be, very easily available because that's a project that's concluded maybe 20 years ago. So we have all the information where the genes are. And now the ENCODE tell you additional information. For example, this RNA track, it tells you where you find the transcripts. As you can see here, you have a peak. That means maybe this gene, I don't know which gene, because the amplification is not enough for us to see the gene names, but you can see, okay, maybe this gene gets expressed highly in this sample because you have a lot of transcript, right? And otherwise, you still have other transcript, but at a relatively lower level. So one gene gets highly expressed. And then if you study this histone H3 lysine 27 trimethylation, that means you have 3 methyl group attached to this lysine. It's a trimethylation to this trimethylation to the lysine residue of histone H3 on the N-terminus. Where do you see the modification? You see peak around this place. So you have a lot of modification around this place. And now you have another track that shows you histone H3 acetylation. So this is acetylation. It's not methylation, it's acetylation. So this acetylation happens in a lot of places, but as you can see here, the level is very low when you compare to lysine 27 methylation. Therefore, you kind of predict maybe these two modifications on H3 indicate opposite information, right? As you can see there, they indicate opposite information. And then you have additional information of DNA's hypersensitivity. It tells you this same region have the lowest hypersensitivity. If a region have the lowest DNA's hypersensitivity, that means maybe that region is more closed, have a lot of nucleosome. And when you put all the information together, I didn't explain everything. Maybe I should explain the light blue or light green part, BFBR also. So the BFBR is just a transcription factor. It's a transcription factor. You can tell the same region here have the lowest transcription factor binding. So when you put all the information together, you probably can start to predict maybe this region that I'm pointing, which have lowest acetylation, the lowest DNA's hypersensitivity, and the lowest regulatory factor binding, and also high modification by a methyl group on the lysine 2017. So maybe this region is closed. It's a closed region. So that's their conclusion as well. So this region probably is a repressed region, while a lot of other regions are more active. So they have done the work. They put a lot of information in their database. We just go there to search, but apparently they can't do all the situations. Like they probably collected 800 normal individuals, but if we are caring about liver cancer, and then the information will be totally different. So we will have to generate that information if that's what we're interested in. But here I show you the example why this epigenetic information is so important, and it gives you a landscape in a particular health state. What's the modifications throughout the genome? So you're not studying just one particular gene. You're collecting the information throughout the genome of that particular patient or a group of patients, so that you can identify hotspot, which region could be contributing to this disease, to the pathology. So remember, we're not doing anything genetics. It's all epigenetics. Okay, so with that, I move on to something that's more relevant to our discussion of stem cell biology. I would like to introduce this concept called active, bivalent, and silenced. So I'm trying to decode the histone code in different cell types. Not me, this scientist have illustrated this, this figure here. On top, that's what happened in some genomic locus in embryonic stem cells. And in the bottom, that's what happened, exactly the same locus. Okay, it's the same locus. We're talking about the same genomic region or same genes, but shown here is what happens in embryonic stem cells, and in contrast, what's happening in neural cells. Apparently, neural cells are differentiated functional cell, while embryonic stem cell is stem cell. So there are two different cells, but we're observing the same genomic region. So here, first of all, what we are trying to study is pluripotency genes. Pluripotency genes, of course, they should be expressed in embryonic stem cells, but not in neural cells, right? So the first example here is a pluripotent genes. As you can see, if you do RNA sequencing, you're going to have a lot of transcript. That means you have a gene expression. And if you observe the modification in the promoter region, you see this green color indicated as shown here is H3K4 methylation, trimethylation as indicated here. It's lysine 4 trimethylation. So you probably can predict, okay, such modification is H3K4 trimethylation indicate that this is a open region. That means the promoter is region. The promoter is open. It's ready to get regulated by transcription factors. Therefore, the pluripotent genes will get expressed. Now let's move on to beyond the promoter. We go to the enhancer. Enhancer are another regulatory region of the same gene. As you can see here, that the enhancer have K4 methylation 1. That means just the one methyl group. Okay, not like trimethylation, you have 3 methyl group. Here it's methylated by just one methyl group. You have K4 methyl 1. And you also have K27 acetylation indicated by the round ball there. It is K27 acetylation. You also have P300 modification. P300 is the enzyme. Okay, so you have all these modifications. You can kind of predict those modifications helping the locus to get transcribed. Okay, now let's go to what happens in neural cells. In neural cells, what you find is the same promoter, the same enhancer is modified completely differently from what's in embryonic stem cells. So in enhancer, you don't have any particular modification. What you see in embryonic stem cells, those K4 methylation, K27 acetylation are no longer there in the enhancer region. And in the promoter region, what you see is a lot of K9 methylation. A lot of K9 methylation. The K9 methylation actually means it's a repressing mechanism there. You see the modifications are different residues. Those two residues are quite close to each other, but they indicate different things. They will suggest different things. The K4 will indicate open chromatin, while K9 methylation will indicate a closed chromatin. Apparently, they're mediating some structural change in a different way. Okay, so that's easy. Basically, in pluripotent stem cells, you have pluripotent genes in open chromatin, or in neural cells, the pluripotent genes are not in open chromatin. Their chromatin are closed. But let's observe some other things. Like in neural genes, so we're going to another locus that encode neural genes. You can see in neural genes, in neural cells, of course, the promoter is modified just like the pluripotent genes are modified in embryonic stem cells. You see this HK4 trimethylation. You also see this K27 acetylation. So that's neural genes. Of course, you can understand that because neural genes are active in neural cells. But look at the neural genes in embryonic stem cells. You don't see actually a lot of negative indicator, right? You don't see that K9 methylation. You don't have that here. You probably have some. Okay, I'm sorry. You have some K9 methylation. You have a K9, not K. You have K9 methylation here, but you also have the active indicators. You also have K4 methylation. So you have a mixture. You have a mixture that region sounds like it hasn't made a decision. Like that region have some positive modification as well as some negative modification. We call that bivalent. So basically, in embryonic stem cells, they don't turn off the neuron genes completely because that's not economy. They don't want to turn it off and later they have to turn it on. That probably will cost a lot of energy. But instead, we put the marks there They put both marks there, negative and positive markers. Therefore, you have a bivalent state. So when the embryonic stem cells later on will differentiate toward neuron cells, they can easily turn off the negative mark. Okay, so that's called bivalence. So the bivalency happens a lot in stem cells. They don't turn off the somatic genes completely. Instead, they make a state that is called ready. They're ready to get those genes expressed. But they put some negative markers, so practically it's not expressed yet, but it's ready. The bivalency means, it's a state that put that genes in a ready state. Okay, and then of course silenced. I won't go there because when we explain that the pluripotent genes is active in ES cells, and of course they're silenced in neurons. So if you're interested, you can go through this and ask me questions. I guess one of the important things is try to memorize some positive and negative modifications, such as K4 modification, which is almost always active. Well, the K9 methylation is almost always negative. Well, K27 methylation is negative and K27 acetylation is positive. So when it is positive, it is shown in green color, and when it is negative, it's shown in other colors. I don't have a text description of the histone code, but hopefully you can study that illustration and help yourself to understand a little better about the content there. But scientists have come up with the term histone code. That means they are trying to put the modifications into categories like I did in the previous slide. Some modification is called active modification, some is called a negative modification that try to turn off the gene. But life is a lot more complicated. Biology is a lot more complicated than that. A lot of times those code won't work. It won't work. I'm going to give you 2 examples. Because in the future, if you are doing this kind of study, if you are a clinical doctor, and you ask a company to sequence, to give you information of a particular group of patients, and you probably will face the challenge, how to interpret those modifications. Which region is indeed a true hotspot that's associated with the disease? It's highly likely you may encounter that kind of challenge. So I'll give you a couple of examples. In the first example, first of all, let me give you an explanation of what happens with the first 2 columns here. So first is a chip, it's OCT4 chip. So OCT4 chip, that means they tell you where OCT4 as a transcription factor binds throughout the genome. OCT4 is a pluripotent stem cell gene. That's one of the factors that Shinya Yamanaka used. So it is a pluripotent stem cell gene. So basically they give you, so in some regions you have a lot of binding, in some regions you have less binding. So they put all the binding regions as shown here. They sort all the binding regions by read density, like from high to low. They also use a macrococcal nucleus. If you still recall, I explained macrococcal nucleus. Macrococcal nucleus prefer to cut around the nucleosome. So basically, macrococcal nucleus sensitivity indicate compact chromatin. This is to the opposite of DNA's hypersensitivity. So here it's very strange. You see that OCT4 binding sites have 2 categories in terms of macrococcal nucleus. So basically, some region doesn't even have any macrococcal nucleus activity, but some region have a lot of macrococoa nucleus activity. That is kind of controversial because, as we know, that macrococoa, if you don't have macrococoa, that means it is open chromatin. So this part of binding is easy to explain because it is open chromatin. In this open chromatin, you have octavore binding. But at the top is hard to explain. As you can see, you have a lot of macrococoa nucleus activity. That means that part have a lot of nucleosome. However, octavore is still able to bind to those regions. And eventually, they found that the octavore binding is different in these two occasions. So in the bottom here, so it's called a de novo nucleosome. De novo at nucleosome depleted. That this is the original binding site of octavore. The octavore can bind because it is nucleosome depleted. Well, the top sequence is nucleosome enriched, and octavore can still bind. And the sequence here, the binding sequence is very different from the, to the original binding site of octavore. And eventually, the scientists were able to find is in this nucleosome enriched region, octavore bind to half site. And somehow those two half sites get put together, maybe by a 3D structure of chromatin. Therefore, somehow octavore can still bind there. You maybe get confused a little bit, but wait until I explain what is a pioneer factor, you will start to appreciate why. So we will predict that octavore bind to nucleosome depleted region, but however, octavore does bind to nucleosome rich region. And then scientists found that these two regions, the binding between octavore and those sequences are different. Octavore bind to different sequences, bind to some so-called half site, not the full site, but half site.
Dr. Liu?
Yes.
Could you explain what exactly does it mean by de novo nucleosome enriched? Because I know de novo means something new or synthesized new, but like, how does that relate to nucleosome enriched?
Basically, the de novo, they don't mean that this is not a true binding. It is a true binding. The canonical is what's already known. The canonical is what's being done elsewhere previously. Without this chipstick technology, we already have known, maybe by just randomly synthesize some nucleotide, and then you try to study the interaction between a protein and a nucleotide, you found that octavore can bind to this canonical sequence, the so-called canonical classic sequence. But now in this study, they call de novo, basically that's what they found in this study.
Okay, gotcha.
Yeah, I think that's, they found in this study, when they compare the two sequence to the classic sequence, you can see that it is the nucleosome depleted region is quite similar to the classic binding sequence.
Yeah.
Only one nucleotide different.
Yeah.
Here, otherwise it's quite similar. Well, in the nucleosome enriched region, octavore bind to something that's totally new.
Yeah. Okay.
Not as what we
usually
knew previously, but they found that it is like a half site. It's an ATGC, and it's just half of the originally identified sequence.
Okay.
But the other half is located far away from another different place.
Oh, okay.
Look at here. It's another sequence. It's half site here.
Okay, that makes much more sense. That makes much more sense.
Okay, so this, when scientists get something that they can't explain, they try to dig deeper to find additional information. And here, that's what they found.
Okay, awesome.
Okay, maybe those two half sides somehow get pulled by the 3D arrangement of chromatin, and now octavore combined to those places.
Okay. Okay, awesome. Thank you.
Yeah, it is. I don't think the details are known, but they have some important discovery there.
Sure.
Yeah, and here I have another example. That is in another paper, what they found is they're trying to do reprogramming. They're trying to make mouse embryonic fibroblasts to become neuron cells. This is a direct reprogramming, not toward pluripotency, but towards another somatic cell type. So what they're trying to do is to find where one of the transcription factors that they used for reprogramming, the factor is called ASCL1, where this ASCL1 will bind in the fibroblast genome. So they're trying to identify a transcription factor binding site in fibroblast. And of course, at the same time, they also, did the landscaping. So basically, they find where those histone modifications are. Okay, that's, so this alignment here basically shows you the 0 means the ASCL binding center, the center of this transcription factor binding site. But they found that the center of that transcription factor binding is also marked by several modifications, like K27 acetylation, the light green color. And then they have this purple color indicating a K9 trimethylation. And then on top, they have a K4 methylation as well. So this 3 major band here is the top 3 modification. That is kind of confusing because I don't think you will be able to recall what I said in the previous slide. I was in that ES and neural cell slide. But I would say this again: the K4 methylation and the K27 acetylation indicate open chromatin. Okay, so the K4 and K27 acetylation here indicating a positive modification, while the K9 methylation is always considered to be negative. It's extremely negative. It is almost never associated with an open chromatin. So that K9 is a very negative modification there. But you can see here this transcription factor binding to a region that is marked by both positive and negative. But that negative is so negative, we don't even consider that to be bivalent most times because the negative is too negative. So the scientists actually get confused because why? Because in this fibroblast, this transcription factor shouldn't be able to bind to this region because there's a hugely negative mark there. And then they did this H3 density staining to see the nucleosome occupancy because they want to really appreciate whether this is an open chromatin or closed chromatin. They use the histone modification as an indicator, but that only is an indicator. It's indirect evidence. So they studied nucleosome occupancy directly. So you don't know whether you have a lot of nucleosome there. But again, they see, okay, you have a lot of nucleosome occupancy there. That means indeed it is closed chromatin. So it is again against our theory because we always think that transcription factor like to bind to open chromatin, not closed chromatin. But in this case, apparently, the fact is that particular transcription factor actually bind to a closed chromatin. So that's lead to the concept called the pioneer factor. The pioneer factor. The pioneer factors are a small group or small number of transcription factors. They don't need the chromatin to be open. So they can open the chromatin by themselves. So that's why they are called pioneer factors. When they go there, they bind to the chromatin in silent state. So basically that place doesn't have any gene that express that. It's totally silenced state, but the transcription factor, the pioneer factors can bind. They don't care whether the chromatin is accessible or not. They will make it accessible for themselves and other transcription factors. So when they bind, we start to make the the chromatin to become a competent state, and eventually they're going to pull the naked DNA out and remove all those negative modifications. So eventually they are going to make that region to become active state, and in active state it has to be open chromatin. So some transcription factors has the ability to go there and bind to the closed chromatin. So that's the concept of pioneer factor. So that's the last. I would say it's the last because I still want to introduce the concept of histone variants, but I guess I will leave that part to yourself. That's another part of epigenetics. So I'll just do a summary here. The epigenetic regulation is is mainly composed of DNA methylation, histone modification, and chromatin remodeling and histone variants. So this shown here is histone modification, and here is DNA methylation, and then this third group is histone variants and chromatin remodeling. The histone variant basically is we have H2A, 2B, H3, and H4, but that's not all. The histones have a lot of variant, called H2AZ, H2AX, more than 20 different variant of histone encoded in different places of the genome. So different genes, but they're slightly different from the classic histone. And whenever you see that the variant histones are participating in nucleosome, that usually means something. It's either DNA repairing or active chromatin or silenced chromatin. So the histone variant, it's not modification. Now it's a totally different, it's not totally different, it's a different protein there. The sequences will be quite different. The modification is on one residue, but now if you call it a variant, that means you have a lot of difference. So those variant histones can contribute to silencing, activation as well. So I have some slides you can study by yourself. Otherwise, I will conclude today's lecture. Just to, I hope you appreciate that DNA methylation and histone modification both contribute to the epigenetic landscape, how genes get activated and silenced. And you have a lot of enzymes involved in the process. You have for histones, you have writers. That means writers will put the modification on histones. You have erasers. The erasers will remove those modifications. And on top of that, you have readers. The readers is different from erasers. The readers will recognize this modification. They see, okay, this histone is not modified. I'm going to bring additional factors here to do a different job, different from the unmodified histone. Okay, those are called readers to recognize the modifications. Okay, that's the end of today's lecture. Thank you, everybody. If you have a question, you can stay behind and ask me.
Thank you, Professor.
You're welcome.